Microbial nutrition, ecology, and growth. The reliance of these microbes to survive based on the select microbial nutrition that helps them to sustain energy. So remember, one of the signs of life is to be able to metabolize proteins, metabolize carbohydrates, metabolize lipids, and metabolize nucleic acid, which is a two-way highway. The first way is to catabolize, break down nutrition, and the other way is to anabolize, build up the nutrition. Okay, so we begin by defining very important concepts that microbial nutrition is every substance that we're able to get as microbes. We're able to get ATP and energy out of. Now you are a nutrition. There are macronutrients and micronutrients. I ask my students all the time, what's the difference between macronutrient and micronutrient? Is macronutrient giant, macro, big, and micronutrient is tiny? No. Macronutrient means you need it in large amounts. That's what macro means. You need it in large amounts. And micronutrients is a nutrition that you need in small amounts. For example, inorganic substances are micronutrients because you need only a little bit of them, like the vitamins. If you have too much of the micronutrients, you're not supposed to, what are you going to get? You're going to get intoxication. You're going to get vitamin intoxication. So they're micro, not because they're small and tiny, they're micro because you only need a little bit of them to survive. You don't need too much. Macro, you need too much of those. If you don't have enough, then that's going to lead to a deficiency. So the acquisition of this nutrition is regulated by processes by which chemical compounds and nutrients are acquired from the environment to sustain life. Your ability to acquire and to exploit, sustain energy out of all of the micro and macronutrients reflects your life. That means you are alive. So what are bioelements? Bioelements are basic requirements for life. So there are certain particular substance known as the CHOMPs. CHOMPs, CHONPS, meaning carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, and now they added to that the sodium, the calcium, the iron, the chlorine, and the magnesium, because we realize that all of these electrolytes are important in your body. In fact, there are diseases where there is hypermagnesemia, hypomagnesemia, hypercalcemia, hypocalcemia. You know, people don't think much of that. What's the problem with having too much calcium? I mean, the more the better. You know, calcium makes your bones stronger. Actually, that's not true because too much calcium is implicated in the heart, in the contraction. So if you have too much calcium, that's gonna prolong the ventricular phase, and that's gonna cause arrhythmia. It's gonna cause a disruption in the heart rhythm, in the heart rate. And if you have too much calcium, that's gonna turn on. You know, some enzymes are supposed to be off. Calcium is a switch that turns them on. Calcium turns them on. So having too much calcium is going to turn on some enzymes that are supposed to be off, and that's going to lead to, enzymes accelerate some particular reactions, and that's going to cause detrimental effects. So these are the bioelements. They are the basic requirement for life. These are substances. We don't make them. They are there, generated in life. We take them from our environment around us. We drink the milk. Did you make the milk? No, you drink the milk from the cow, you drink the milk from the goat, and the acquisition of these nutrients from the environment makes them biomolecules and bioelements. We discussed essential nutrients. Essential meaning you don't make them yourself in your body. You have to get them from outside. That's what makes them essential. Yes, they are important, but not only are they important, they can never be brought except from outside. Your body is unable to make them. And we have macronutrients. These are required in large quantities, and these are proteins or carbohydrates. And there are micronutrients, and these micronutrients, or sometimes we call them trace elements, because you need only a trace, a little bit off. And those are the zinc and the manganese and the nickel. And you know, those are your prosthetic groups. Those are part of the enzymes. Remember, hemoglobin has heme prosthetic group on it. Hemoglobin has iron group on it that is, required for the activity of that particular enzyme. Sometimes some medications, all we need is to destroy the prosthetic group. The enzyme is not working, then the microbe is going to be dead. What are organic nutrients? Organic nutrients have to have both carbon and hydrogen. Students think that organic molecules only need to have carbon. Well, what about carbon dioxide, CO2? Carbon dioxide has carbon, but is it organic molecule? No, organic molecules need to have at least one of both, carbon and hydrogen. You're absolutely organic. So our proteins, nucleic acids, lipids, carbohydrates, these are the four big macromolecules of life. Are they all organic or some of them are inorganic? No, they are all organic. If you remember the structure of what a sugar looks like, it has a lot of carbons and hydrogens, the sugars, the sucrose, the monosaccharides, the disaccharides, the polysaccharides, they all have carbon, they all have hydrogen. In addition to that, if you look at the amino acids and proteins, don't they all have carbons and hydrogens? The lipids is made up of glycerol and free fatty acid. It's a lot of carbons and hydrogens. Nucleic acids have a lot of carbons and hydrogens. Yes, they can have on top of that, on top of carbon and hydrogen, they are welcome to have any metal. They can have phosphate, like in the DNA, there is a lot of phosphodiester bonds, right? Deoxygenated ribonucleic acids. There are sulfur groups in the disulfide bridges of proteins. But that doesn't matter because we do have at our core substance, we have carbon and hydrogen. So all of the molecules of life are organic. In fact, there is a theory that says all of the organic molecules, we love organic. Organic means that it has carbon and hydrogen. All of the organic molecules came from inorganic molecules. So who was there first? The inorganic molecules were there first. And then from them, we were able to develop the organic molecules. Very good. So an example, methane, like we just saw there. If you see there where my mouse is, organic molecules, we got all four. Carbohydrates, lipids, proteins, nucleic acids. They're absolutely, all of them are organic. They're welcome to have some inorganic on top of them. It's okay, but they are all organic.
So the methane is in terms of organic part. I'm not understanding where methane.
So methane is an example of a substance, an example of a substance that is a perfect example of an organic substance because it's made of purely carbon and hydrogens. That's the smallest type of organic molecule. One carbon atom, and the carbon atom valence shells allow it to have 4 bonds and is bonded to 4 hydrogens. That's the purest, smallest form of an organic molecule. And then he showed you all of the organic molecules we have in life: carbon, hydrogen base, carbohydrate, lipids, proteins, and nucleic acid. Inorganic nutrients. These are atoms or molecules that contain a combination of atoms other than carbon and hydrogen. So now you see something like magnesium sulfate, sodium oxalate, calcium oxalate, sodium phosphate, sodium chloride, the salt, the white death salt. Sugar is also another form of white death. Sometimes they exist in form of gases. So we have oxygen, carbon dioxide, and water. So if we analyze the chemical structure, the composition of the cell, we see that the cell is bathed in aqueous solution. It's made up of 70% of water. And if we dry the water in the cell, we see that 97% of the dry cell weight is organic molecules, because that's where all the money is at. That's where all the ATP is. When I perform catabolism of lipids, catabolism of carbohydrates, nucleic acids, I'm going to be able to get a lot of ATP. So remember we said all these organic compounds are composed of 6 main elements. Call them carbon, hydrogen, oxygen, phosphorus, nitrogen, and sulfur. So we refer to them as, you make your own mnemonic, but I just say chomps, and it reminds me if I say CHONPS, chomps. Chomps is carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Those are the main constituents that make our organic compounds. So organic compounds that can never be made, they can never be made by an organism because they lack the genetic and metabolic mechanism to synthesize them. So there are growth factors that must be provided as a nutrient for survival. So for example, we need essential amino acids. If you deprive some particular bacteria from the essential amino acids and vitamins, they're going to die, which is one of the ways we rely on the pharmacological interventions to kill the bacteria by depriving it from the food or by shutting down the metabolism of bacteria for food. Even if bacteria was able to acquire and steal food from you, it is no longer able to metabolize it. We shut down all the metabolic activity with our drug or medication. We shut down bacteria's ability to catabolize all of these organic compounds and make energy out of them. Bacteria is known to live and adapt to live in crazy places. So there was an oil spill. There was like a back in the day, like the politics now has changed. For example, you cannot build a factory in the middle of the city. It's not allowed anymore. No matter how much money you have, they're never going to approve it because they know there's a lot of dangers with the pollution, a lot of dangers that come with, all these chemical toxins. Back in the day, like about 100, 150 years ago, there was a huge toxic waste spill from one of the factories. It spilled out of the factory and it fell into a lake, and it poisoned the entire lake, killing all the bacteria, killing all the trees, killing all the plants, the algae, the animals. So it was a disaster, and that is what led to making laws that you can put factory next to natural life, because if some accident happened, you destroy the whole net. It's not easy to bring life back. We save life, but you cannot. If life dies, you cannot make life. So after a while, they took a sample from that deadly oil spill in that lake that has now turned into, they call it the lake of death. So they took a sample from the lake of death and they wanted to analyze and see, what are these toxic wastes. And guess what they found? They found bacteria in there. They found that some bacteria is able to live in that lethal. That's not life form. That is anti-life. They are living in hell. So that shows you how bacteria was able to adapt. They even now propose that there's some bacteria living in the sun. In the sun. That's how hot it is over there. Some bacteria adapt to live in there. They also discovered that some termites. You know what termites eat? Wood. Wood. They eat wood. They found some bacteria living in the digestive system in the intestine of the termite, eating wood. I mean, this bacteria is nuts. So now it shows you the ability of the bacteria to acquire, and that's the reason in their strength is their simplicity. They're not like us. We are so complex. We have a multicellular eukaryotic structure. We have the plasma membrane, we have the nucleus, we have the cytoplasm, we have a lot of demands. People from the city are very complicated people. People from the rural areas, they are so simple, they are satisfied with anything. Like the dinner for them, like they milk the goat and you drink the milk. The Amish, yeah, they drink the milk and that's dinner. For you, is that dinner for you?
Hell no.
No, you want a nice dinner with the appetizer, and you want this, and then the dessert. They will go crazy if you think like that. But some bacteria are so simple. That's why if there's a big problem happen with the famine and poverty, who's gonna survive? The Amish or us? Yeah, the Amish, because they're used to living in a very, very austere type of life. That's the same thing with bacteria. They can live in the sun, they can live anywhere because their demands for life are so primitive and simple. So, what are the main determinants of the nutritional type? So, there are carbon, we classify our organisms based on the energy source. So, some bacteria are heterotrophs. So some organisms are heterotrophs. Heterotrophs means that the organism must obtain the carbon in an organic form, like protein, carbohydrate, lipid, nucleic acid, made by other living organisms. So this microbe, it can never survive, this microbe, it can never survive unless you give it, it's like you need like steak dinner and you need all that kind of stuff, carbohydrates, nucleic acid. Steak has all of it. It has lipid, it has the nucleic acid. So when you eat the tissue made of cells, so the DNA of the steak, you're going to use it to replenish your own nucleic acid DNA. And then also you're going to need the sugars. So you always have like some pasta. But some other type of microorganisms, they don't even need organic molecules. They have nothing to do with whole foods. So they like inorganic molecules, meaning they consume carbon dioxide and they consume sulfur and stuff that we can never consume. So you see this, we love organic molecules, carbon, hydrogen. They don't want that. They want inorganic. So that's an organism that uses carbon dioxide or some inorganic gas to survive. So if I tell you on the exam, I have a type of a microorganism that relies on organic substance, you absolutely know this particular organism can only survive if we give it proteins, carbohydrates, lipids, and nucleic acids, then that's a heterotroph. Call that a heterotroph. Autotrophs, those are the ones that rely on the opposite, inorganic. There's another classification. There are chemotrophs. Trophs mean, so the chemotroph, they gain energy from chemical compounds. So we use chemical compounds, whether organic compounds or inorganic compounds. In the end of the day, if you use a chemical compound, organic, inorganic, we don't care. As long as you use a chemical or a substance, then you are a chemotroph. And a phototroph. Photo, exactly. Photo is a photo, light. So it relies on the sunlight energy. There's different types of energy. Chemical energy is not the only type. Your gas combustion, when you go and you fill your car with gas, that gas, is gas organic or inorganic? No, it's very organic. Just because you can't eat it, it doesn't mean it's not organic. People think organic is only when you eat it. No, it's very organic because this carbon hydrogen bond, when you burn it, it emits a lot of energy. So chemical energy is a form of energy, like a Bunsen burner. That's a form of energy. But when you warm yourself, You converted the chemical energy into heat energy. So now these guys now, they don't want chemical energy. They want light energy. Everybody, they want their own energy. Yeah. You have a question?
No, I'm saying the sun, right?
The sun, yeah. So they get it from the sun. So there are different types of energy. And that's what makes this life really interesting. And that is, how did they
So what
make...
show mean? Okay.
So troph means a user. A user. So use chemical substance or light source or a light source. Okay, so that is a very interesting chart because it shows us. This shows us the category or the carbon source, where we get our carbon from. And then we have, remember, we have two classifications, two trophs. One troph for the carbon source. Are you taking carbon that is organic, or are you taking carbon that is inorganic, carbon that has no hydrogen in it? Or then we have another separate classification, and that is the energy source. Are you relying on photo, light, energy, or are you relying on chemical, chemo? So if you look at the autotrophs, carbon dioxide, let's look at the first example. Photosynthetic organisms, like the algae, the plants, and cyanobacteria are a special type of bacteria that is just like the plants. They are a special type of bacteria. They are just like the plants in their ability to rely completely on photo energy. Now, if we look at some other certain bacteria and archaea, like these methanogens, methanogen, you could tell from the name, methane, so that means they are organic or inorganic?
Organic.
Very good, they are organic. And some deep sea bacteria, those guys are, so now we cannot call them photo. We cannot call them photo because they don't rely on light, they rely on methane. So why they call them methanogens. So these are chemoautotrophs. Now, when we look at the heterotrophs now, they rely on organic. So an example of that is look at the fungi, the protozoa, the bacteria, the animals. We have chemotrophs, and they convert nutrients that they obtain from other organisms. So there are the saprobes. Saprobes metabolize the inorganic matter from the dead organisms. They're very important. I mean, these microorganisms, the pathogenic microorganisms, are actually, even though they are harmful, thank you, we need them. Imagine these saprobes. Remember saprobe. Remember that. Saprobes, they metabolize the organic matter from dead organisms. So when you have a dead body, so now, let's say, God forbid, all of us right now, we died, and they leave us in this room. If they come back a week later, this room is going to smell terrible. If they come back a month later, a little better than before. 2 months later, 3 months later, 6 months later, it's not going to smell like anything because the bacteria removed these saprobes. Those are the ones who are responsible for they're responsible for metabolizing all the dead bodies. So now that's how we have space in this world. Imagine everybody who lived before us from when the earth was made up to now.
Then it would be filled with a bunch of dead bodies.
Exactly. So that's one of the usefulness of them. They are important. You know, you can't say, I wish I lived in a world where there's no bacteria. Oh, you don't, because you're going to be dealing with so much dead body.
It's so crazy. It's still filled because you have some people get buried underground. Mm-hmm. They still, and everybody else had the body is still there. So it's like, you know, so that's kind of taking up space.
No. Once you bury somebody, the earth changes. The earth is, you think the earth crust is just the way it is, just staying like that. It looks stable from the top, but in the bottom is moving. Like these guys, they think like somebody's 7 feet under. They go in front of the grave, and he's gone. He's gone 100 years ago. He's moved away. That's how Earth is. That's how life is. They keep moving down until you're. I never figured out where they end up, but they're just not there no more. They're gone. They just keep moving away. And you go to the grave, you stand in front of it, and there's probably like 700 different people that is.
But wouldn't the casket still be there? Casket.
The casket is gone. You know, they eat wood too, the
But
termites.
they still be having that sense.
But don't they make like metal and stuff?
They still have that, you know, the stone.
Yeah, but that's only at the top. That's only at the top. And it takes a while. So like when if you try to dig a grave that's 100 years old, the casket is gone.
But wouldn't the bone be?
Yeah, the bone, it's gonna not, because they don't break bone. But after a while, the bone is just gonna collapse.
Oh, it's gonna like this.
Yeah, it's gonna just collapse. The integrity with age, like with many
Yeah,
years.
it has to be a lot.
Yeah, a lot of time. So eventually you're gonna become dust, and then you're just gonna be gone after a while. And then you're gonna come back up in a form of plants or so, that kind of thing. So, but your soul is gonna be like there. Your body is gone.
Reincarnation, you'll be in a whole other body.
I don't know, but I know is your body is gonna be gone, but your soul is not. The bacteria cannot get to your soul. Your soul is gonna be waiting for the day of judgment. Anyways, symbiotic microbes. So they obtain organic matter from living organisms. Yeah, but you're not gonna come into another person's body, or you're not gonna come into a lion. Yeah, that's not gonna happen. Yeah, that's not gonna happen. That one I promise you all. You're not coming back here again. You're going there. Where there are you going? Heaven or hell? That's different. But you're not coming back here. You're not coming. Nobody's coming back here. Yeah.
I'll be a student in your class again.
Over there? I don't know if they do that kind of stuff there. Over there, no more work. It's just heaven or torture. I hope I go to heaven. I hope we all go to heaven. Okay, symbiotic microbes. That word is one of the best words. Symbiotic. So, symbiotic is when we're both working. My favorite symbiotic word, that what made me like love science back, I will never forget this. They were like in 3rd or 4th grade, like, you know, back in the 60s, like, anyway, when I was really young.
You're not that old.
Okay, so I saw the teacher was telling us a symbiotic relationship is when we both gaining advantage. An example of that would be when the crocodile or the alligator. I never figured why you guys here call it an alligator, but back there we call it a crocodile.
It's 2 different animals.
2 different animals?
Crocodiles and alligators are 2 different animals. They look very closely. You know, look at the gene classification. They're 2 different things.
I'll look into that. Interesting. So one thing that the alligators or the crocodiles do, they open their mouth so wide. They open their mouth very wide. And what happens? The birds will come and pick and nitpick all of the food that the crocodile, like a toothpick. It will remove all of the food from between the teeth of the crocodile or the alligator. Everybody's happy. So the birds got food and the crocodile got a toothpick. But anyways, that's another example of symbiosis. And it actually happens. But actually in your body, you have some bacteria that is in your intestine, living in there. Doing what? They colonize your gastrointestinal tract, and they fight any other bacteria that comes into your body. They're not fighting it so they want to defend you. It's just a conflict of interest. They are fighting all the other bacteria because that's our place. This is where we get our nutrition, from the leftover food that this person eats. You're going to come here, you're going to compete with us. So they end up acting like your immune system. So that's another example of a symbiotic relationship where nobody's harmed. In a symbiotic relationship, nobody's harmed. Both are winning. We are winning because they are like our immune system fighting other bacteria for our sake. Even when you get really old and your immune system gets weak, the bacteria doesn't get weak. It's there fighting for you, defending you. So that's a symbiotic relationship. But the opposite of that is a parasitic. Now that's the problem.
That's what you can use.
Yeah, the pattern is one is plus gaining, the other one is minus is losing. Now that's an example when the bacteria infect your body. They are winning, they are eating your flesh, but you are suffering. You are suffering. So the symbiotic microbes, they obtain of organic matter from the living organisms, and we have photoheterotrophs, whether are sunlight or organic matter, and there are purple and green photosynthetic bacteria. They are completely reliant on both. They are reliant on light energy in addition to chemicals. So the transport movement of substances across the cell membrane. There is passive transport and active transport. Passive transport does not, that's why they call it passive. It's like you're sliding downhill.
A lazy river.
Yeah, it's like you're sliding down a river. And you're sliding, but there is still an energy that is driving that force. It's called the energy of bombardment, the energy of heat, and the energy of kinetic energy. Do not be like the students who think that passive transport requires no energy. That's not true. Passive transport does not require chemical energy in the form of ATP. That's correct. But it does require another form, and that is kinetic energy. You know, the energy of bombardment, people bumping into each other in a subway, when people getting out the subway or getting in. Sometimes you feel you're moving without even walking because you're getting pushed around. That's exactly the energy for diffusion. If you have a lot of particles on one side, they push each other, and then they move from where? From an area where there's a law, from an area where there's a high concentration to an area where there's a low concentration. So that's diffusion. What is osmosis? Osmosis is a special property that water has. Water is a versatile solvent. So we also understand the properties of water. So what makes water, the properties of water. What makes water the substance of life is number one, water is a versatile solvent, meaning that if there is a competition between all the solvents out there, who will be the number one in being more likely to dissolve any particular substance compared to the others? It's water. Water is a universal, versatile, or universal solvent. Number two, water has high specific heat capacity. Water has high specific heat capacity. Number three, water has cohesion properties. And number four, water has adhesion properties. So water being a versatile solvent through the property known as osmosis. So we just discussed diffusion, and we said that what happens in diffusion. By the way, this chomps, don't write this chomps. Tell me what chomps stand for. C stands for carbon. H stands for hydrogen. O stands for oxygen, and N stands for nitrogen, and P stands for and S stands for sulfur. Diffusion is when particles move from an area of very high concentration of solute particles to an area of low solute particles. Is this movement going to happen eternally, forever? Diffusion means diffusion is the movement of particles from area of high concentration of particles to area of low concentration of particles until we reach equilibrium. So for example, if I have 500 millimoles of glucose on one side and 300 millimoles of glucose on the other side, and I allow for the movement through a permeable membrane that allows for the passage of glucose. What do you think is going to happen now? The movement is going to be from 300 to 500 or 500 to 300?
500 to 300.
500 to 300. Is it going to move forever?
No.
No. Only 100 is going to move to 300. Now it's going to be 400. And the 500 is going to lose 100. And that's going to be 400. They are both equal. That's equilibrium. That's diffusion, when the actual particle itself is moving. Now, if you look into this same exact example here, where you have glucose on two opposite sides, one is 500 and one is 300, but the pores are a lot tinier than before. So if you look now, you see that if you look now, you will see that there is very, very tiny particles. It's not like before. So glucose can never move from an area of 500 to 300, 100 moves over now until both are equal. That's not going to happen because the pores are too tiny. So what's going to happen now? The water, instead of glucose, the water is going to move from where to where? From 500 to 300 or from 300 to 500?
From 500 to 300.
From 300 to 500, because water wants to dissolve where there is a lot of particles. So the water, so now it's going to look like that. Water is going to go down at the 300 level, and it's going to go up at the 500 level until it dissolves it. Why? Because water is a versatile solvent. That's known as osmosis. This is known as osmosis. Osmosis. Osmosis is the movement of water from an area of low concentration, exactly the opposite of low concentration of what? Can you say low concentration? So low concentration of water?
Yeah, so water.
No, it's not less. Low concentration of solute particles from 300 to 500, where there's 300 of glucose who cannot move. So we will see that an area of low concentration of particles to area of high concentration of particles. Are we clear? Questions? Now, another thing that you need to know, in your cell, what if you have this problem here? You have a lot of glucose on the outside, and you have a little bit of glucose on the inside, but glucose cannot pass through your plasma membrane for many reasons. Number one, glucose is so big, glucose is huge. Number two, Glucose is polar. You know the plasma membrane is non-polar. The plasma membrane is lipid and glucose is polar. Number two, plasma membrane is small, glucose is big. So you're still gonna diffuse, still glucose is gonna move down, but it's gonna need a facilitator. So that's called facilitated diffusion. In facilitated diffusion, you have some big machine that will literally grab big old glucose and move it down concentration gradient. So we still diffusion. Where's diffusion? Right here. We still movement of glucose from an area of high glucose to area of low glucose until we reach equilibrium. The only difference is what? We need a carrier, we need a facilitator. That's why we call it the facilitated diffusion. We cannot diffuse without a facilitator. If you have 1,000 people on one side of the river and nobody on the other side, if there is no boat facilitator, you're still gonna move from where there's a lot of people to where there's a little. But you need a facilitator to carry you. So this carrier is called facilitated diffusion. Okay, bulk transport. So that is all passive. So all of these different types of transport are reliant on chemical energy of bombardment. You know, there's so many particles, they're pushing each other, so they move from an area of a high particle to an area of a low particle. Water diffuses because water is a versatile solvent. Water has high specific heat capacity. What does that really mean? Water has a high specific heat capacity.
Can you warm it up on a very high temperature?
The boiling point?
Yeah, it has a very high boiling point. Why does water have a very high boiling point? You know, ammonia weighs as much as water. Water is H2O, two hydrogens, one oxygen. Ammonia is NH3, one nitrogen, three hydrogens. They weigh the same. Oxygen and nitrogen, they weigh very similar, and two hydrogens on oxygen and three hydrogens on nitrogen. They both similar. But nitrogen is in the perfume. You spray it, it evaporates. Even room temperature is too hot for it. It makes it evaporate. While good old water needs 100 degrees Celsius for it to evaporate. That's why there is volcanoes in the bottom of the ocean and water is not evaporating. So why is water able to evaporate much later and resist so much heat compared to ammonia that weighs the same? You are asking me. I'm asking a question, and you're asking me. If you don't know the answer, if you know the answer, raise your hand. If you don't know, I'll tell you the answer. Okay, it's because of the dotted bond. What's the name of the dotted bond? Let's say I have a solution of ammonia, 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 like that with three H's. Ammonia with three H's. Exactly. So now if you have oxygen with two hydrogens, oxygen with two hydrogens, and an oxygen with two hydrogens like this. So let's now look at a solution of liquid ammonia and solution of liquid water. Ammonia is going to look like that. Each ammonia particle is on its own. But when you look at the water particle, it has covalent bonds like that. But in addition, on top of the covalent bonds, because oxygen is so electronegative, oxygen is so electronegative, it's putting all the electrons, making hydrogen electropositive, hydrogen electropositive, hydrogen electropositive. Now you see there are interactions. There are dotted bonds between positive hydrogen and negative oxygen, positive hydrogen and negative oxygen. In ammonia, only dashed lines, no dotted lines. In water, there are dotted lines. These are the hydrogen bonds. And those are the ones that make the water stick together like so strong cohesion. So If you boil this, those guys are gonna, they say when you put the sticks together, it's hard to break them. When they're separated, it's easy to break them. But that's exactly what happens with ammonia. They are separated from each other, so they don't have hydrogen bonding. But the water molecules, they're bonded to each other, so it's very hard to break their bonds. So you need a lot of high temperature, so that's high specific heat capacity. In addition to that, there's cohesion, so they're holding on to each other. An insect can walk on water so easily. But if you have an ammonia solution, or if you have an alcohol solution and it's colorless, the insect thinks it's water. When the insect steps on the surface of the alcohol solution or steps on the surface of the ammonia, it's going to fall and drown and die because they don't have cohesion. Adhesion. What does adhesion mean? When you stick a piece of glass into the water and pull the glass out, the water will be sticking to the glass. If you dip the glass in alcohol or in ammonia, it's not sticking to it. And this property, we use it in our body, where your heart has a balloon full of water around it that's adhesive, sticking to the heart. The lung also, the pleura, the heart has the pericardium. You study that in anatomy and physiology. All of this thanks to the cohesion, adhesion, high specific heat capacity, being versatile solvent, is because of the water hydrogen bonding. Active transport now requires, you see, I don't like the way that the book is saying it. The book is saying that it does not passive, let's edit that. It says passive transport does not require energy. That's not true. It doesn't require chemical energy in the form of ATP, but it does require kinetic energy, kinetic or heat or bombardment energy. Now, the active transport requires chemical energy in the form of ATP. Okay. Okay. Diffusion and molecular motion. That shows you the net diffusion of molecules down their concentration gradient by the random thermal movement. You see thermal, heat, bombardment movement. And we talked about this, the diffusion of water from an area of, you see, all the water is moving to where the solid particles are. So some cells, the effects of differing osmotic content solutions. So you see isotonic solution is a solution where the water inside and outside are the same. So now, if you examine this particular, let's look at 3 cells. Let's look at 3 cells. All the cells have 300 milliosmol on the inside. That doesn't change. Milliosmol. 300 milliosmoles. You put cell number 1, cell number 2, cell number 3 in different solutions. You put cell number 1 in another 300 milliosmol solution. So the cell is 300 milliosmol, and the solution that we are bathing it in is 300. So that's isotonic. Because the inside and outside is the same, that means the water moving inside and the water moving outside is equal. It's isotonic. Now, if you put the 300 in 30 milliosmoles, well, that's interesting. Now the cell has more concentration of particles inside than outside. Is the movement of water inside going to be more, or is the movement of water outside going to be more? Who can help me? The movement of water now, if you put 300 milliosmol in 30. Yeah, so the water is going to go where? Inside. It's going to go from where there is less movement of water from an area of low concentration here to an area of high concentration on the inside. So that leads to the swelling of the cell, known as turgor. So the solution is hypotonic. You're right. Solution is hypotonic compared to the cell. So it's going to turgor, it's going to swell, and it's going to burst the cell. That's what happens to marathon runners who are running. They're over dehydrated. They're hot. Now the blood osmolarity dropped, and the red blood cell osmolarity is still high. So when they get to the finish line, they drink the water and drink and drink, and water keep going inside the cell until they have so much water inside their body. Now the water keeps on going inside the cells till the red blood cells burst. Many people die every marathon. They will never tell you that. Yeah. When they get to the finish line, you drink a lot. So you have to be moderate. So if you drink too much water, your blood is going to be so dilute, hypotonic, then the water is going to start moving into your red blood cells, bursting them. So now you have no red blood cells, you can't get oxygen, no aerobic respiration.
Or it's like an IV. If I stand here and I put 5 IV back in my arm, eventually. It's like an IV. If I stand here and I put 5 IV.
But the IV, no. The IV is not pure water.
Oh, it's salt water.
Yeah, it has some substance. Yeah, not only salt, sometimes glucose, it depends, and that is gonna keep preserving the osmolarity.
But sir, if you don't have integrity, don't it will move out because the body is very unique.
But you can't drink too much
If you don't what? Can you say it again?
If you're getting too much water, can't it leak out of the intracellular into the extracellular?
If you have too much, if you drink too much water, can that what?
I'm saying when the body recognize it and then move it out of the cell and once it goes somewhere like in the extracellular.
No, because when you're drinking so much water, the digestive system responsibility is let me deliver the water from the intestine to the blood. That's its job. Now the water keeps coming to the blood, and it's coming at an overwhelming capacity to that your kidneys cannot take the water into your urine. Because you're drinking too much water, far exceeds the capacity of the nephron to filter that water into the urine. You keep on dumping water because you're so thirsty, and the water now the blood is bathed in so much water. So now the osmolarity of the blood drops, and water moves from the blood into the red blood cell and bursts. Yeah, so your blood is drowned, not your body. Your blood is drowned in water. Okay. Hypertonic solution, that is when the water now is going to move from the cell to the outside, to the tissue. That would happen in dehydration. When you're so dehydrated, water there's no water is evaporating in the sweat, in the saliva, so the water is being removed from your body at a high pace, so the osmolarity of the blood is going to increase now so much because the water is going out. So now then it's going to shrink. It's going to cause, so here you have 3,000 milliosmoles, water keeps moving out, and that's going to cause shrinkage.
So is that a result of dehydration?
Yeah, because when you're dehydrated, meaning you have not enough water, so now the acids start to build up, and you have a lot of waste, a lot of substance. They used to be diluted with water, but it's no longer there. Okay, that shows us the three we talked about. Those facilitated diffusion, we discussed that. You see the facilitator, see the elevator. We still obey the law. The law is you still move from an area. Remember when I told you, you have 2 islands, there's an ocean between them. You have so many people on one side, few people on the other side. You're still going to move from where there's a lot to where there is a little, but you need a boat. You need a facilitator. That's why they call it facilitated diffusion. Carrier-mediated active transport. Carrier-mediated active transport is when you now, that's your sodium potassium ATPase, is when you bind to the outer, like for example, we hydrolyze ATP in order to move. So this is carrier-mediated active transport. I am using gas energy to move something. Active transport is not like passive transport. Passive transport is a slippery slide. Active transport is going back up that hill again. So the same thing now is I'm using chemical energy to press the gas so I can climb the mountain with my car. But the facilitated diffusion is when you climbing, when you're sliding down the mountain, which is not really that challenging. So imagine, in Black Friday, people are running over each other to go inside the store. Now imagine, actually that's an example of diffusion from where there's a lot of people on the outside trying to go where there's a little bit of people on the inside. Now imagine they have the rock, the rock. The rock is standing at the door. And if anybody runs in, he will grab them and throw them back out against everybody who's trying to come in. That's the sodium potassium ATPase. And the sodium potassium ATPase is interesting. It's like two of the rocks because it's doing two things. It's taking because there's a lot of sodium on the outside and a lot of potassium on the inside. So the natural movement is for sodium to move in and for potassium to move out. The sodium potassium ATPase does two jobs. Every 3 sodiums that come in, it's going to throw them out. And every 2 potassiums that move out, she's going to bring them back in. So it needs a lot of ATP in order to do that. So endocytosis and exocytosis. Endocytosis from the name, endo. So you see we make a bleb, we make a balloon, we make a vesicle that is internalizing and bringing substance to the inside of the cell. So that is phagocytosis, where there's an ingestion of substance or the cell. So in pinocytosis, there's an ingestion of fluids and dissolved substances. These are known as the microbial. So pinocytosis is water drinking or cell drinking. So you see that the oil substance or the water substance is being uptaken by the microbial. But phagocytosis is when the white blood cell is crawling with pseudopods and walking closer to the bacteria, engulfing it and swallowing the bacteria. Now, what are the environmental factors that influence microbes? Is the niche, number one, the environmental niche, that's the totality of adaptations or organisms make to their habitat. That's an example of that toxic waste spill, and you still have some bacteria living in it. And factors include temperature, oxygen requirement, pH, osmotic pressure, and barometric pressure. You have some bacteria that are able to adapt to extremely high temperatures, and some are able to adapt to extremely low temperature. Some bacteria are able to live in very high oxygen rich places, and some of them are living in very low oxygen environment. Some of them live in acidic, very low pH. Other bacteria live in very high pH, alkalos. Some are suffering from osmotic. Some live in places where there's high osmotic pressure, and others are living in places where there's low osmotic pressure. And barometric pressure, that is the pressure that increases. So air pressure, does air pressure increase as we ascend the atmosphere or we descend the atmosphere? Think before you answer. What happens to the pressure? When you go up, does it increase, or do you go up when you decrease?
You decrease.
Raise your hand if you think pressure increases as you ascend the atmosphere and go up. 3 people, 4 people, 5, 6, 7. Okay. And raise your hand if you think the pressure goes up if you go down. 1 person.
Me. I
Okay, two people. Okay. You see the submarine exploded in the air or exploded in the bottom of the ocean. You know they wanted to go see Titanic? Huh? Why? What's funny?
Nothing.
Yeah, the people when they moved, when they tried to get on people tried to get on the submarine and they wanted to go see the Titanic deep in the bottom of the ocean and exploded because the pressure is so high as you move down.
But what about when you get on the airplane?
When you get on the airplane is a pressurized is a pressurized cabin. So there's pressure in the cabin because there's the pressure up in the air is so low.
That all the pressure is.
Yeah, so you think that that's the air is not the air, it's the cabin that's creating that artificial pressure for you to breathe. Because if the pressure is so low as you move up in the air, you're not going to be able to breathe because you need for air to be high so air can go from where there's high pressure, like diffusion of air, from high pressure to low pressure. But when you go up in the air, the pressure is so low, oxygen is not even moving into your lung anymore because the pressure is so low. So
they drop the oxygen mask.
yeah, the oxygen mask is to supply you with oxygen, but we're not talking about the oxygen. We're talking about the pressure who is guiding the movement, the energy that allows the air to go from the oxygen tank into your lung. So that is important as far as the boiling point also. You know, you want to cook the best meal, cook it in a submarine. If you want to make the best spaghetti or you want to make the best, if you like boiled chicken and you add spices to it, or like you want to cook something, why is it best to cook it inside the submarine?
It's gonna be tender.
So, yeah, but why is it tender? Because you have, let's say somebody's in a mountain, somebody's at sea level, and let's say that's the sea, and this is a submarine inside the ocean. So somebody's trying to cook up here, somebody's cooking at the sea surface, somebody's cooking in the submarine. So obviously the pressure here is the lowest. The pressure here is the sea level, and the pressure in the submarine is the highest. Now there's a relationship between the boiling point and pressure. So the boiling point is corresponding to the pressure. So if the pressure is low, the boiling point will be low. So if the pressure is high, the boiling point will be high. If you try to cook at the top of the mountain, people who live on the top of the mountain, water will evaporate long before the spaghetti cooked because the boiling point drops for water. The boiling point for water is 100, but when you're on top of the mountain, the boiling point is becoming now 70. So when the water heats up only to 70 degrees Celsius, the water is disappeared. The spaghetti is still hard. It's not soft. It's been a while since I had spaghetti. I'm gonna go get spaghetti. Where do they get that from? The pizzeria?
Pizza shop. Yeah.
Pizza shop.
Or you can make it yourself.
That's too much work. Anyways, the submarine now, that's the best one because you're so deep in the bottom of the ocean. Now the boiling point for water at the sea level is 100 degrees Celsius. But deep down in the submarine, it's gonna become like 300 degrees Celsius. Imagine you're cooking spaghetti or you're cooking chicken at 300 degrees water boiling. It's not even like you don't even need an oven. You just heat. It's gonna be done fast. It's gonna be cooked really thoroughly. So as you go down in the submarine.
So you need to make
Oxtail. People, raise your hand if you ate oxtail.
It's good.
So I'm the only, okay, raise your hand if you did not eat oxtail ever.
You never had oxtail?
I know you want to say something to me.
No. What's wrong with oxtail?
Why have you never had it?
I only say things about pork. Oink, oink, oink.
I pork now.
I ate pork the other day.
Because of what?
I got second guess pork now because of what I learned from you.
It's not from me, it's from science.
Well, I know, but
I'm a messenger.
The fact that you said
Okay.
it and me really thinking about
The fact that I'm teaching, I didn't say nothing.
Oh my God, like you get it. Like, I'm saying
Okay, thank you. Anyways, let's go to, all right, adaptation temperature. we discussed minimum temperature, maximum temperature. No matter where you put oink, 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 temperature is not gonna die. The eggs. Did I tell you guys about the parasites living in your body? And the more you eat, excuse me, the more you eat sandwiches like chicken, and the more you eat beef and all of that, you know they live in on our digestive tract. So they eat with us. So the more you eat that kind of stuff, the more they're gonna survive. The more you become vegetarian, or the more you like fast, they die quickly. So now you're gonna have a really clean, and you're gonna have a really like pure digestive tract because they haven't had food for a while, so they die quickly. No. That's what I wish, but no. Okay. As oxygen is utilized, it is transformed into several toxic products. So that's now oxidative stress. Oxidative stress meaning that there's a lot of free radicals, especially with the metabolism of alcohol. You see, people that drink the alcohol, this alcohol once, you know, it looks good outside in the bottle, in the glass. That's how they get you. They get you with the way that it's bubbly, it's bubbling. But once it goes into your body, it turns into snakes. It's eating you from the inside.
Snakes.
How is that? Because the alcohol is metabolized to acetaldehyde.
Yeah.
So there is an increase in the oxidative stress, and there's a lot of electrons that are free radicals, and these free radicals are increasing the oxidative stress, damaging the cellular structure, causing necrosis and causing inflammation. And one of the major diseases that is caused by oxidative stress because of alcohol consumption is pancreatic cancer and acute pancreatitis. There was a very big scientist, was a brilliant scientist, and the problem, he was just an avid drinker. He used to drink a lot, and you know he was diagnosed with pancreatic cancer. He knew that it was because of it, and you know everybody came to his last final lecture in the school, and the whole place was packed. And he was trying to tell people, you know, don't feel sorry for me, I'm okay. And he was doing push-ups, but in the end he died because of all of the excessive oxidative stress that is coming from the unhealthy lifestyles, and especially with the consumption of the alcohol. So we have a system of detoxification.
Yes, sir. Those who don't drink it and have the pancreatic, what science says is one of the other?
Well, then we know that the main reason for causing pancreatic cancer is the alcohol consumption. Some people get it, but it is not. I don't know why. I don't know. But all I know is that the main reason that causes it, like 90% of the people, there's 10% of people who get pancreatic cancer that is not alcohol related. It is related to something else. What is it? I really don't know. Sometimes it's idiopathic, but what we need to know is that we already have a built-in system in our body to detoxify all the waste, and to neutralize and mute any electrolytes, or mute any electron, or shut down any oxidative stress. But when the lifestyle is very unhealthy and there is a consumption of the alcohol, then that is going to is going to severely overwhelm your bodily healthy systems that are there to counter and to mute this stress. So they're showing you now the main types of oxidative stress inducers, and that is the singlet oxygen and the superoxide. There's an enzyme that is helping you. It's called superoxide dismutase. That's our best friend because it mutes any superoxide that comes. In fact, this superoxide can sometimes be helpful to us because your white blood cells use the superoxide in order to kill any bacteria or parasites. So all of these superoxide dismutase, the peroxidase, okay, these are enzymes. So superoxide, dismutase, peroxidase, those are ones to save us from the accumulation of hydrogen peroxide or the accumulation of superoxide. But we use those, and we use those, not the enzymes, we use those free radicals to kill parasites, bacteria, because they are part of our white blood cells. So the weapons that the white blood cells use to kill the microbes, to kill the bacteria, to kill the viruses, to kill the fungi, to kill the parasites, are those same ones, are those superoxides, those single oxygens, metalloproteinases, degradative enzymes, and acid that burn all of the microbes and the bacteria. Okay, now when it comes to adapting to oxygen, so oxygen is important for aerobic respiration. So we are obligate aerobes. What does obligate aerobe mean? It means that you're obligated to get oxygen. If there's no oxygen, you're dead. But some bacteria, they're known as obligate aerobes. They cannot grow without oxygen. They're just like us. Some of them, they're like the crocodiles. They can live on the sea. They can live in the water. Those bacteria we call facultative anaerobes, meaning they utilize oxygen, but they can also grow in its absence. They're like the crocodile. These are called the crocodile bacteria. They are facultative anaerobes. If there's oxygen, they'll take it. If it's not there, they'll survive too. And then there are the microaerophiles. These require only small amount of oxygen. Small. Aero, oxygen, file loving. They love only a little bit of oxygen. They don't need too much. Then when you look at the anaerobes from the name, anaerobe, meaning that it doesn't use oxygen. Obligate anaerobes, they lack the enzymes to detoxify oxygen, so it cannot survive in an oxygen environment. Can you believe that? Oxygen kills them. They are obligate anaerobes. They don't want even a little bit of it. Just like there's some obligate aerobes, we need all oxygen. They need none of it. And then there are the aerotolerant anaerobes. And from the name, you can tell they're aerotolerant. They don't need oxygen, but if it's there, it's not going to kill them. They're going to survive. They utilize oxygen, but they can survive and grow. I'm sorry? They don't need oxygen. They are anaerobe. Look at the name. They are aerotolerant anaerobes. So their nature is they are anti-oxygen. They are anaerobes. But they're aerotolerant. So if there is some oxygen, it's not going to kill them. It's not going to choke them.
Like the little water turtles?
Turtles?
Yeah.
These are bacteria. I don't know the turtles. Yeah, turtles are like the...
They can live in water, but like they can tolerate.
Yeah, but those we're talking
you're
here.
Talking about that.
Yeah, we're talking about anaerobes who can tolerate air. And there are some also facultative anaerobes that utilize oxygen, but if it's not there, they can do without it. It's okay to them. Okay, so now that's the trick we use against them. That's how we tell which kind of bacteria do we have. Nobody knew what kind of bacteria is aerobic, so they did all the tests. They gave some oxygen to some bacterial culture. If it all died, they knew that this is an obligate anaerobe. They took some other form of bacteria and they gave it some oxygen. Then they removed oxygen. If it survived both, that shows it's facultative anaerobe. So there are cultures with reduced media that contain oxygen removing chemicals such as, so you need to know that thioglycolate is whenever you put thioglycolate, you're going to remove a lot of oxygen. Oxygen is gone. And that can help determine the oxygen requirement of a microbe. It can tell us the more you put thioglycolate, the more oxygen is removed. So we can see how much of that we can add, how much of thioglycolate can we add, and the bacteria is still around. So if we add a lot of thioglycolate and the bacteria is still alive, you can tell that they are anaerobes because we pretty much moved all the oxygen out of their metabolic pathways. So the relative position of growth of bacteria that differ in oxygen requirement in such culture media is going to provide some indication of their adaptations to the oxygen use. So now we move on to pH. We finish with oxygen. So the majority of the organisms grow at pH between 6 and 8. This is a special range you need to know, that between pH 6 and between pH 8, this is the playing ground, the playing field of all of the bacteria. There are some neutrophiles. They grow on a neutral pH, and that is the range of 5.5 to 8. That's the neutrophile. Acidophile, you can tell from the name, acidophiles, those love acid. So they grow at extremely low pH. And alkalinophiles, those love alkaline pH, they love basic pH. You see, bacteria developed a wide range of oxygen demand, a wide range of temperature, a wide range of acidity. So the osmotic pressure, you see that most microbes exist under hypotonic and isotonic conditions. Interesting. They don't live in a lot of hypertonic conditions. So that's why people believe in like spilling the salt or sprinkling salt. Actually, that's only a little bit true, because when you sprinkle salt in any table, that's gonna make the medium hypertonic. So bacteria do not like hypertonic mediums. They only like isotonic and hypotonic mediums. So there are osmophiles. Osmophiles require high concentration of salt. These are halophiles. So they have, in order for them to survive, they need a high concentration of salt, while the obligate halophiles, they grow optimally in solution of about 25% sodium chloride, but they require 9% of calcium salt, not very salty, like salty lakes and ponds. Examples of the osmophiles are the halobacterium and the halococcus. Then we have osmotolerant. So you know those tolerant bacteria, they do not require high concentration of a solute. And we have facultative halophiles, and these are remarkably resistant to the salt. An example of that would be Staphylococcus aureus. Now, when we look at the pressure variable, we finished with the salt, we finished with the osmolarity, we finished with the acidity and the oxygen demand. Now we're looking at the pressure. There are barophiles. Those love and survive extreme pressure, and they will rupture if they're exposed to normal atmospheric pressure. They will only live at extreme high pressure. So where will you find those? Exactly. So they'll be found with in the bottom of the ocean with fishes. And when it comes to water, they're only dormant at dehydrated stages. Like you see these spores and cysts. That's what I was talking to you about earlier. When we eat the steak, we eat the chicken, there is a lot, because I told you, we're not the only ones who love meat. So all of the bodies of the cows, the lambs, the goats, they have a lot of the cyst, and they have a lot of spores. Spores and cysts, they are like a crucible. They are a shield. It's like an adaptive mechanism to protect the bacteria and help it evade the immune system attack. When the immune system mounts a lot of antibodies and mounts a lot of white blood cells, they are hiding inside the shell, inside the spore, and that will protect them. So when we're cooking the food, some of them get burned and killed, but some of them are not. So now the people who develop the lifestyle of they don't eat a lot. They just like when they're so hungry, they eat like like figs or dates, or sometimes people drink the milk, and they have like pumpkin or a vegetarian type of a vegetarian form of food. Those people, they have a very clean digestive system because the worms, the parasites, the cysts, they don't survive on that kind of food. So they will be so hungry they will die. But when you eat a lot of the food, the meat, and you have, that's why on the pyramid of the hierarchy of diet, you see a lot of salad and vegetables in the bottom or at the top. On the bottom, because that's the base. So your food should be 80%, 90% salad, and 10% of the chickens. Because now, because remember, every time you're eating chicken and beef, you're bringing in a lot of cysts. But when you bring in those cysts, you already had ones already in your digestive system, and now they're sharing the milk with you, and they're colonizing more and bringing in more and more parasites. So you will have irritable bowel syndrome. Okay, that's not what I'm trying to do. I'm trying to go over the concepts. So you see that a lot of the food is really dictating your nerve tension. So people with irritable bowel syndrome, they will be so calm when they eat like food that is friendly and they don't eat the food with like a lot of cysts. But when they have like a meal with steak, then there's a lot of irritation because all of these parasites are now active and now moving. So bowel syndrome, yeah. Yeah. So sometimes it's also genetic. It's not you can't say somebody, oh, you have irritable bowel syndrome. You've been eating a lot more meat than you're supposed to. It's not only about that. It's also genetic, but it gets aggravated a lot when they eat, the red meats and that diet. So you need to appreciate that a lot of microorganisms are living in the stages of cyst and in the dormant spores. In symbiosis, there are two organisms that live together in a close partnership. So we have mutualism. We talked about mutualism, where both members benefit. So they're obligate mutualists. They require each other to survive. Non-obligate mutualism, they can be separated and they live apart. Commensalism, you need to know the concept. Commensalism means that there's commensal benefit of the members neither harmed or benefited. So one type benefits while the other type is really not affected.
Just coexist.
So your coexisting means both of us is like a mutual, coexistence. So I benefit from you and you benefit from me. We're both benefiting each other. But commensalism, one is benefiting while the other one is really not affected. They said that there's a fish that is always accompanying the whale and living like on the side of the whale, and and they eat like the dead meat at the fins of the whales. That really doesn't benefit the whale with anything, but it's really not harming it. So that's an example of commensalism. Parasitism, that's the worst, when the parasite is dependent and benefited at your expense and harming you. So that's an example of these ecological associations. They would obligate mutualism, where organisms are intimately associated, they require each other to survive, and non-obligate mutualism, organisms interact at cellular level. Discuss that. Commensalism. Okay, now we have non-symbiotic organisms. So when organisms are free living in a shared habitat, there's two types of non-symbiotic relationships. There is a syntrophy, known also as the cross-feeding, and there is the amensalism. Syntrophy is when microbes are sharing a habitat that is feeding on the substances released by other organisms. But in the amensalism, one member of the association is going to produce a substance that harms or kills another. So that's an antagonistic, while the other one is synergistic. It's helping and supporting the growth of the nearby organisms. Syntrophy. Example of syntrophy in ecological associations would be microbes sharing a habitat that feed off the substances released by other organisms. Examples of that, the azotobacter releasing the ammonia, and it feeds another nearby microorganism the cellulomonas. And these organisms degrade cellulose and they feed and return back. So it's like a cycle. So like us and the plants. The plants take the carbon dioxide, give us the oxygen. We take the oxygen, we give them the carbon dioxide. That's a synthropy. So we both are using metabolites that and relying on each other. Amensalism is one member of an association produces a substance that harms and damages and kills another. What are biofilms? Biofilms result when organisms attach to a substrate by some form of extracellular matrix that binds them together in complex organized layers. So that's very interesting. We have these films that are made when microorganisms are attached to a substance by some form of extracellular matrix, and it's going to bind them together in complex or organized layers. They dominate the structure of all of the natural environments on Earth. So we see that these bacteria live in communities known as the biofilms, where there's a lot of communication. They communicate and cooperate in the formation and the function of these biofilms. So what are the interrelationships between the microbes and the humans? We discussed already that the microbiota is the sum of all the microbes that are living in our digestive tract and they interact with us. The human body is rich in a habitat for symbiotic bacteria, fungi, and a few protozoans that are the normal flora. These are the normal flora that you need. Sometimes you take probiotics in order to increase the normal flora because they are there to help you. So microbial growth occurs at two levels. There's growth at a cellular level with increase in size and increase in the population. And there's a division of the bacterial cells occurs mainly through binary fission. So the parent cell enlarges and it duplicates its chromosome and forms a central transverse septum. Binary fission is how they divide, just like the mitosis of the bacteria. See, the chromosomes, first of all, replicate, and then you have the fission where there's an closure completing the cell compartments, and there's not a lot of organelles to duplicate. There's not a lot of DNA to duplicate. So binary fission is much more primitive and much more rapid when compared to mitosis and cell division, because the cell cycle is reliant on mitosis with all its stages, in addition to the interphase, the G1, the S phase, and the G2 phases. So the rate of the population growth, you see that the time required to complete the fission cycle is called the generation. So the bacteria develops exponentially. So one generation gets doubled. So if you see that the number of the cell grows exponentially, so each new fission cycle is going to increase the previous population by a factor of 2. So the generation times vary from minutes to days. So in the laboratory, populations typically display a known predictable form of and pattern of exponential growth. And there are known stages known as the lag phase, exponential growth phase, and then there's a stable stationary phase, and then lastly is the death phase. So that shows us the lag phase, when the bacteria is beginning to exponentially increase. Then we have this hike, the jump, where there's exponential, massive amount of bacterial growth, and then there is a stationary phase where bacterial growth is going to be stable, and then the bacteria is going to undergo the death phase, and then we're going to end up with the end of the cycle. So another cycle begins all over again with the lag phase, exponential phase, stationary phase is an oscillation. So methods of analyzing the population growth is we have the turbidimetry. That is the most simple mechanism. So we look at the degree of cloudiness, the turbidity of the nutrient culture media, and that shows the population size. So we utilize a spectrophotometer where we emit light through a glass prism to diffract the light. So now we have separated the light based on wavelength. So when the light is passing through a tube, and the tube has a more exponential population growth of a particular bacteria, and you subject the light to pass through it, do you believe the absorbance is going to be higher or the transmittance of light is going to be higher? If there's a lot of population, it means that the absorbance is going to be high because the bacteria is going to absorb the light and not going to allow any light to pass through. But if the population growth is so weak, you will see that the transmittance light, the transmittance percentage is much higher because there's not a lot of bacteria, there's not a lot of bacterial products, these metabolic products, they're not being built up in the tube, they're not being built up in that environment, so they're not absorbing the light and the light is passing through. Okay. And that's how we count the bacterial cell. There's a direct cell count, usually using computers, and we count the number of the cells in the sample microscopically. We count the cells in direct count, and they can be automated by the sensitive devices. These are the sensitive devices that count actually for us. That is the flow cytometer that counts for us actually the number of the bacteria that is inside the tube. Literally count one by one how many bacteria grows in there, which is very interesting. Any questions? Okay, please